class is about molecular biology, and so we are going to talk about some of the fundamental molecular biology experiments that were used to discover a lot of the material that I'm presenting in class. And so one of the fundamental and classic experiments that elucidate DNA replication was the Michelson-Stahl experiment. And so what they did is they grew up E. coli in heavy nitrogen and then isolated their DNA. And so if the E. coli were exclusively had heavy nitrogen, they would be heavier. However, if they were grown in regular nitrogen, they would be lighter. So in order to answer the question of are E. coli, does replication occur in a semi-conservative fashion? They grew the E. coli up in this heavy nitrogen for a couple generations, and then they shifted them into media that had light nitrogen in it for one generation. And so if they had seen conservative replication, what they would have seen is that after one generation in the light nitrogen, you would have ended up with two populations of E. coli. One that still had the parental heavy nitrogen DNA, and the other that had the now light DNA daughter strands. But what they saw is that after one generation grown up in the light DNA, the light nitrogen, all of the bacteria had shifted to having a mixture, a sort of half and half of the heavy and light nitrogen. So the question is then, what would have happened if they had grown up the E. coli for a third generation in the light nitrogen? So think about this for a little bit. What would have happened? So you would have had two parental strands that have been now divided into the sort of light heavy combination, but after a second generation grown in this light nitrogen, you would have now started developing alternative bands. That is, bands that were only light nitrogen, as opposed to some of the bacteria that were still a mixture of the heavy and light. If you